Can you hear me?
Well, I can hear you.
Awesome. How are you doing today?
Good. How are you?
I'm doing okay. Last couple of days I felt a little frazzled because I got a lot going on, but yeah, everything's going all right.
I'm running for student senate. I'm putting together a cabinet right now, and so it's been crazy. We have 10 days to put it all together.
So what exactly do you do on Student Senate?
So it's kind of like the governance of like the student body. So being able to, we have a budget of like around $3,000 to give back to the students. So being able to like create Jewel into a better place for students and the community. So just trying to think of different ways to improve the college. And so last year we came up with an idea of like making it like a community closet for people in need and different things. So we just kind of create a list of ideas and then implement it in that school year.
So is the role of Senate going to be any different because you're at a college in a totally different world than you were a year ago?
So I have no idea. I could see meetings being on, Zoom. I could see our budget being cut. I can see several different problems arising. So we'll see what happens once we get involved, but I have no idea what it's going to look like.
Okay.
Yeah, so
Well,
it could be very interesting.
well, welcome to the World COVID Club.
Yes.
All right. Yeah. So I've been a little frazzled because the lab that we normally do in my biology class that I teach in the fall has to be different because we're sending everybody home after Thanksgiving. They're not coming back and they're not going to be here to finish the things that they normally would have started and then come back from break to finish. And so me and the guy that I teach with, we've been kind of troubleshooting the lab and trying to keep it as much like the old lab as possible, but recognizing the shortened semester. And so we were getting feedback from our students in the course of them doing observations in the field and things like this about things that the students were kind of interested in. And so like last week, we totally shifted what our plan was. And so we've been running around trying to get things together at the same time that we had a test in that class on Monday, just like I had a test in this class on Monday. So also trying to get a test ready for a different method of testing, et cetera, et cetera. We feel like we're running around like chickens with our heads cut off. So we feel your pain.
Yeah.
So how's everybody else doing? What was that? I didn't catch that, whatever it was. All right, as we get started, how did you guys feel about the exam on Monday? Just go ahead and unmute yourselves for the time being. Hello. How did you guys feel about the exam?
I felt fine about it. There were a couple of things that I guess I had missed in the study guide.
Okay.
I was not sure about.
They were in the study guide and they appeared on the test and they were in the study guide and you just didn't pay enough attention to them, or they were on the test but they were not in the study guide?
I'm not entirely sure. The one specifically that I was thinking about was the corn walking question.
Oh, okay. Yeah, we did talk about that. That was all in the introduction kind of to why we went into such detail about photosynthesis. So that was, it's difficult sometimes to keep us rooted in the omnivore's dilemma while also covering a lot of science. So whenever we're going into something sciencey, I always try and introduce it by saying, here's the section in the omnivore's dilemma. This is why we're about to go on this like 4-day jaunt into photosynthesis. So yeah. And those will probably necessarily be relatively brief references back, so they might fly past you without you noticing them. I'll try and do a better job of calling attention to that when we're going through it. What else about the test?
I kind of ran out of time at the end. I just felt like I didn't have enough time, maybe just a little bit more, but it was all right.
Okay. All right. So one of the things about the way I write questions is I generally have a specific answer in mind. And a lot of times on short answer tests, students want to just like vomit out everything that's in their head on that topic. But I try and make the questions relatively focused. And you'll see, I didn't post a key yet, did I? No. All right, I've written the key and I have a digital version of it. I'll post that. When you look at the key, my guess is that all of your answers are 4 to 5 times longer than my answers are. And so really pay attention to the question, read the question, and once you've answered the question, you can just stop and move on to the next question. I am not actually interested in knowing everything that you have stuffed in your head about a particular topic. I'm usually interested in 1 or 2 particular things about that topic that you have stuffed into your head. And that will actually save you some time. And you can always quibble with me after the fact with you get something wrong because you didn't write enough and you're like, well, the question asked for this and this. And I'm like, oh yeah, the question did just ask for that and that. So yeah, that might help later on.
Yeah, I'm glad. Yeah. I'm kind of glad you said that because this has been like the first test. It was just, I just didn't know what to expect, honestly. So I'm glad you said that.
Yeah, I have the same difficulty with my biology students. I ask a question about natural selection and then they just blurt out whatever they have in their head about natural selection. And really I was just looking for one little response. So
Yeah.
All that critical thinking education.
yes, think a little critically during an exam. Was it weird taking it in a digital form, or did that not bother you?
I liked it. I think it allowed to take us like out of like the noise of like the classroom and be able to focus like on our own like knowledge and not have to worry about like competing necessarily with other people, but we were able to focus on like our own education in a way.
Yeah, there's usually like a tension in the air in a classroom during a test.
Oh, okay.
For whatever reason. I think it's because everybody's just stressed about how they're going to do. And sometimes it affects. And taking it at home, there's not that kind of stress in the air.
Oh, that's interesting because somebody asked me about when I was an undergraduate, one of my fellow students in a class that I was taking said, you do really well in this class, like what's your secret? And I had lots of things that I could have said at the time. I said, well, I view college as a competition and who gets the highest grade wins. And she said, you're twisted. So I was actually a person as an undergraduate who kind of thrived on that like in-class competition, but I can get why not everybody is like me. so well, good. As long as it didn't throw you too much. I didn't know how it was going to play out, and I actually learned some things from doing it with you that I applied to a test that I gave later in the day. So what I do is I set a time for the beginning of submissions, which is the beginning of the class. I set a time for the end of submissions, which is the formal end of class, and then I set a later time after which Moodle won't accept it at all. And I didn't know how Moodle was going to deal with that. And what happened was if you turned it in a couple of minutes after class formally ended, it saved it as a draft, but it saved it there. It's there. I saw it. I got it. I've already downloaded all of your tests onto my iPad so that I can grade them digitally. So I wish I had known that going into it because I would have warned you about that. So don't worry. I have all of your exams. It appears that they got saved all correctly and things like that. It's just that if it came in after the due date, it was saved as a draft, and then there was this later time later in the day when it would just stop accepting them at all, because if you hadn't submitted it by like 11:30, then something's seriously gone wrong. And one student had trouble submitting at all. He just came over to my office and we figured out a way to get it submitted. So sorry about the little bit of stress there at the end. I warned my biology students later in the day that this was going to happen if they turned it in kind of after the due date. And I think that led to a little less stress for them there at the end. But now we know that that's how it works, and so we can chill on the next iteration. All right, any other comments? Okay, then I want to do a couple of other things before we get to kind of material.
I have a question about the food blogging.
Oh
Do
yeah.
we turn it in? We turn it in tonight, or no? Thursday night, or we turn in Thursday or Friday morning.
So it goes from Friday to Friday. So you have all day Friday to get your food blogging done.
But do we fill out Friday or no?
What's that?
Do we fill out Friday's journal? Or we
Yeah.
just turn it
So there are two things. There's the food journal, and there is the food blogging.
Oh,
So the food journal.
Food journal is what I was asking about.
Oh, food journal. Yeah, food journal. Just either submit it electronically to me, submit it electronically to the assignment. Did I put the assignment on the Moodle site? I don't remember if I put it up there, but I'll get it up there today.
Do we turn it in this Friday or next Friday?
It's now this Friday because we're 2 weeks.
Mm-hmm.
2 weeks into the process, yeah. So it'll be this Friday. So either submit it to the Moodle site by the beginning of class on Friday, or drop it off at my door. It'll be outside my door for those of you who are turning it in a physical form. On Monday, you can pick it up and then start the process all over again.
Thank you.
Is that clear, Blake?
Yeah, thank you.
Okay, cool. All right. I am going to share screen. All right. Can you see a PowerPoint slide presentation on your screen?
No.
No, I shared the wrong screen. All right. Stop sharing. Let me move you guys to that space and do this. Okay, now can you see it? PowerPoint? Yes. Okay, good. All right. So one thing, there are a couple of things that I've put up on the Moodle site. So if you go to Moodle, you'll see today's PowerPoints, which we'll get to here in a little while. And you'll also see that there is a National Academy of Sciences genetically engineered organism report. So let me go to Moodle real quick. And so if you're in the Ecology of Food class, you'll see that I've added things to this week, the September 23rd materials, which is today's stuff in this Academy of Science report. If you go up to the syllabus and schedule section that is on August 23rd, it was posted the first day of class. We're actually pretty on schedule. And so I want to call attention to a couple of things for you. One of them is opening. So if you look, my computer for some reason is running really slow this morning, and I'm not sure why. I already restarted it once. Oh my gosh, so slow. I had this open earlier, but then I had to restart my computer. Well, that is unfortunate. Sorry about this, guys. Like I said, I had this open actually early in anticipation of going through this with you. Anyway, do you guys have the schedule? Can you guys open the schedule on your own computers? Oh, there it is. It's coming up. So you're going to see, so we just took exam 1. Then we have an introduction to genetic modification. That's what we're going to be talking about today. We're going to talk about genetic engineering. We're going to talk about what genetically modified organisms are, the way people talk about them. We'll talk about the history and state of them. And during this time, what we're doing is we're building the basic building blocks for you to understand this report by the National Academy of Sciences. And so on Friday, October 2nd, what I'm going to have is I'm going to break you guys into 3 different groups. And each of you is going to prepare a short presentation summarizing one of the chapters of this National Academy of Sciences report on genetically engineered organisms. And so right now it's tentatively scheduled for October 2nd. We can move that around if you're needing a little more time and things like that. We'll just reword the schedule a little bit because it's just a couple of weeks. But after today and Friday, you should be pretty well equipped to handle most of the things that are in there. And so it'll be a group presentation. It would be nice if it wasn't too wordy, if you did as much of the presentation through images and then your own verbal explanations as possible. And if you look in the syllabus, you'll actually get a grade for your performance on that presentation. I'll give you a rubric for kind of what I'm looking for in that presentation. But the presentation is going to be based on this National Academy of Sciences report. Do you guys know what the National Academy of Sciences is? Anybody? Nobody? Nobody knows? Okay, that's all right. You're not scientists. You don't need to know what the National Academy of Sciences is, except that they sometimes do things that impact the lives of ordinary Americans. And so the National Academy of Sciences is this body of scientists that you get elected to, and you get elected to it by your peers. And generally, you only get elected to it after you've received an international level of acclaim and recognition for the scientific work that you do. There's also a National Academy of the Humanities, and so similar sorts of thing in the humanities. And one of the things that they do periodically is they look at what the pressing issues of the day are, and they basically solicit a group of scientists to write a report that, in this case, is about genetically engineered crops. And so this was published in 2015. So if you took Ecology of Food before 2015, you didn't have to read this report. And if you took it after 2015, you did. Sorry for you that they put it out in 2015 and you have to read it. So the National Academies it's basically made up of the best scientists that the nation has to offer. And so one of the things that you'll see can you see the report on your screen? Yeah, and you can also download it from the Moodle site as well. But here at the beginning, there's a committee on genetically engineered crops, past experience and future prospects. These are the people who wrote this document. And it's super long, and I'm not going to require you to read all of it, but I'm going to require you to read sections of it. So these are all the people, and one of the things that you'll notice about, what is one of the things that you noticed about the people on the screen? These are all National Academy members who contributed to this report.
Most of them are collegiate professors, it looks like.
Yeah, most of these people are academics. And so these are people at universities or occasionally at think tanks, but most of them are university scholars. You have academy staff. There's a board that kind of was the board that oversaw the production of it, but they didn't necessarily contribute to it. And then there is a food and nutrition board, once again, that also advised them. And then here at the end, well, where'd the reviewers go? Somewhere in here, there was a list of reviewers. I might have scrolled past it. So they write this report and then they send it out for review, just like you would with any scientific writing. And the reviewers look at it and they critique it and they say, oh, well, you kind of got the science wrong here, or oh, are these conclusions really warranted given what is known about this topic at this time? And so let's go. Oh, here's the reviewers. So here are the reviewers. And once again, what do you note about the reviewers as well?
They're collegiate.
Yeah, many of them are at universities. Occasionally you have people that work in industry, you know, somebody from the IBM Corporation, where did I see it just a minute ago? Donald Danforth Plant Center, that's actually a research center that is funded by Monsanto. So there's a mixture of both academics and industry people in that list of reviewers. And so what we're going to do is we're going to basically read through 4 of the chapters. So what I want you to start reading as soon as you can get around to it is start reading chapter 3. Chapter 3 deals with genetically engineered crops through 2015. So it was published in 2015. What this will do for you is it will give you an introduction to kind of the history of genetically engineered crops in the United States and the world. It will also introduce you to the methods by which we have done this in the past and are continuing to do this. We're going to be covering some of that material today and Friday in class. And then what I'm going to do probably later on today, and I want your opinion on this, there are going to be 3 groups in the class. One group is going to focus on chapter 4. One group is going to focus on chapter 5. And one group is going to focus on chapter 6. And so the reason I say that you're going to focus on it is that you're going to focus on it in terms of you're going to do your best to understand that chapter as fully as you can, because on October 2nd, or whenever we decide that we've allowed sufficient time for you to prepare for it, you're going to take over my screen and you're going to present a presentation basically outlining what you think the salient points are about, in the case of chapter 4, the agronomic and environmental effects of genetically engineered crops. And so what they did was they basically reviewed all of the information that there is available on what the effects are on agriculture and the environment in terms of the effects of genetically engineered crops. Chapter 5 looks at human health effects, and chapter 6 looks at social and economic effects. And this actually in the past has turned out to be one of the most interesting chapters for students because they hadn't necessarily thought yet about the fact that these genetically engineered crops might have positive and potentially negative effects on the farmers doing the growing of it, and also having effects on the economics of the broader society. So that's actually, students have expressed to me in the past that that's been a surprising chapter for them. So that day in class, because we have about 60 minutes, we're going to give you about 10 or 15 minutes to make a presentation. Shouldn't probably take more than about 10 minutes. And then we'll have discussion among the members of the class. So everybody will unmute yourselves and we'll just kind of talk about what that group presented. And then when that group is finished, we'll move to the group covering chapter 4, and then we'll move to the group covering chapter 4, then chapter 5, then chapter 6. So my question for you is, would you like me to just randomly assign you to a particular chapter, or would you like to get together with your colleagues and choose what chapters you would like to work on? This is the time I need feedback from you.
I think random's fine. I don't think it'll make much of a difference.
Okay.
That's my opinion.
All right.
I'm good with random.
Okay.
Yeah, I'm probably better just because I don't know a lot of them.
Okay, all right, yeah. I didn't know how it was going to work out, given that we haven't been meeting together in class. You probably don't know one another quite as well as if we had been meeting as a group. Then I'll just randomly assign you, and I'll post those assignments on Moodle later on today. Everybody should read all 4 chapters, but for the preparation for the presentation, you should concentrate on the chapter that you're assigned, because ultimately what we're going to do with this is later in the semester, you're going to write a position paper. And the kind of structure of the assignment is you go home for Thanksgiving, which all of you are going home for Thanksgiving because you're not allowed to stay here. And you are at Thanksgiving dinner, and so there's food on the table, and people are talking about food, and one of your relatives, I'm assuming that this is going to be an elderly, curmudgeonly uncle, says something about genetically engineered crops. And regardless of what he says, it's filled with all sorts of misconceptions. And your job is to write a reasoned outlining of what's known about genetically engineered crops to your uncle, which means that you need to write it in ways that are not going to offend him too much, because it's always good to maintain family relationships. But the other thing that you have to do is you have to explain it to him in a way that it will be accessible to him, knowing that he does not have a degree in biology, and so he's not going to be able to relate to it if you use a bunch of jargon and things like that. So it needs to be a common language document that is maybe in the form of a long letter to your uncle, basically saying, you know, you said some things at Thanksgiving, and I've been learning this stuff in college, and here are some things that I think you ought to know about the pros and cons of genetically engineered organisms in our food supply. And like I said, I'll give you some guidelines for this as we get closer to that date. I envision that happening like actually around Thanksgiving, so that you have some more time to kind of digest the material that is in the report. It is a National Academies report. So there's a lot of science in there. And so one of my goals between now and then is to equip you as much as I can to understand the things that are in the report so that you can write intelligently about the report. So as you're going through there, each day, even if genetically engineered organisms isn't the topic of a discussion, if you're having questions as you're going through and reading this thing, ask them in class, because you will benefit from the answer, and everybody else in the class will benefit from the answer as well. So that's that. Are we clear on what the task is? Okay. All right. So that's
Where
that.
where do we find this at?
Is
it on Moodle? That just a link?
It's on Moodle. So I had Moodle up a minute ago. Let me go back to Moodle. So if you look at the Ecology of Food site, if you go down to today's date, there's so this is the September 20th to 26th week. Here's today's materials. There's a National Academy of Science GMO report. It's in that folder. So if you click on that folder, you'll see the report right there. It's a PDF, and you can just download it. And what I do is I mark it up on my iPad generally as I'm taking notes on things that I read. But you can do whatever your habit is to do with literature that you're reading. All right. So that is that. All right. So let's start going down the path of understanding what we mean when we talk about genetically engineered organisms. Let me see. So a lot of times people use genetic engineering and genetic modification as being the same thing, but the way scientists should use it, and I'm going to do my best to stick with this, is there are all kinds of ways of genetically modifying organisms. One way you can genetically modify organisms is just through selective breeding. So what I've written on the board for you in preparation for class today is 2 different ways that we can think about genetic modification. In the case of Mendel's peas, he had tall plants and short plants. When he bred them together, he got an F1 generation that was heterozygous, and they were all tall plants. And then he bred them again, and he got a mixture of tall plants and short plants. He got 3 tall plants to every short plant. Well, one of the ways that you could genetically modify peas so that you always have tall plants is to just only allow the tall plants to breed, and over time you would have a population that was more and more made up of tall plants and had fewer and fewer short plants in it. Another way of doing this is in what we call additive genetic traits, which we talked about at the end of genetics, where the traits are governed by the small contributions of lots of different genes to that trait. These types of traits lead to these normal distributions of trait value. So we have very few short people, we have very few tall people, and we have a lot of people who are in the middle. The way that we study this genetics is through calculating heritability, where you look at the phenotype of the parents and the phenotype of the offspring. The slope of that relationship is heritability. You can use that heritability estimate. You can apply selection to your population, and then the population will respond evolutionarily. This is how we get beef cattle that are bigger and carry more weight on their bones. This is how we get more milk production out of cows, et cetera, et cetera. This is called additive genetic variation. That's what heritability is measuring. This is dominance variation. This is variation that is due to these genes that have a kind of on-off sort of effects, all or none sort of effects. But you can still select for those traits as well. And so that's the form of genetic modification that people have been practicing with domesticated varieties of plants and animals for 12,000 years. That's different from a genetic engineering where we basically take a gene out of one organism and we put it into the genome of another organism. Or we use a technique by which we take a gene that is in an organism and we either turn it off or turn it on, or kind of turn up the dial on how much that gene is getting expressed. And so genetic engineering is genetic modification, but not all genetic modification is genetic engineering. Does that make sense? Nod your head yeses if that makes sense. Okay, all right. So I want to first talk about kind of traditional forms of genetic modification to kind of set the stage for how genetic engineering is different. So I see memes on the internet. Everybody puts out memes about all sorts of things. And there are a surprising number of memes about food. And so this is a meme that I saw, a visual guide to identifying genetically modified corn. Non-genetically modified corn on the left and genetically modified corn on the right. Put on your critical thinking caps. Everybody unmute yourself for a minute. Put on your critical thinking caps. What are your thoughts about this meme?
I feel like it's just flying over my head.
Okay, that's fine. Anybody else?
I'd say the size of each.
I'm sorry, what was that? Branson?
The size of each. How
okay,
non
so
is
you have,
very small.
this is a tiny thing, and this is an ear of corn that you would buy at the farmer's market. Sweet corn. You guys like sweet corn? Yeah, I like it too. I put it on my tacos. I make street tacos periodically, and that's one of the ingredients. Anybody else have comments about the meme?
On the left, is it actually corn?
It's a type of plant called teosinte. And it is the evolutionary precursor. It's the evolutionary ancestor from which corn was developed by people in Mesoamerica.
Just looks like a bunch of peas sticked on a kebab.
It does look like that, doesn't it? So it differs substantially from modern corn, but it's the evolutionary precursor that led to the development of corn. Anybody else have any thoughts about this meme?
That is inaccurate.
Okay, Mackenzie, why is it inaccurate?
You're talking about two different types of corn, and the corn we're used to seeing is the one on the right.
Okay.
And no matter, even if it's genetically modified, the size could be affected, but the overall look is not going to be affected.
Okay, so when we use the term genetically modified, how did we get from teosinte to modern corn?
Natural selection of the number of kernels and the size.
Yeah, if you're a farmer in Mexico, 9,000 years ago, and you find teosinte and you find that you can eat the grains from teosinte, and you start planting teosinte in your patch of ground that's there, in the Copper Canyon region or wherever, and you notice that some teosinte have more kernels than other teosinte, you're probably going to plant those teosinte because they're producing more corn kernels, more kernels of grain, and eventually you might be able to apply enough natural selection to that thing that it eventually does turn into this. Well, as it turns out, in the same way that we took wolves and turned them into pugs, why? I do not know, and took teosinte and turned it into corn. Much of this, well, in the case of dogs, all of it was through selective breeding, through what we think of as artificial selection, but this traditional form of genetic modification by which we selectively bred organisms. Well, corn developed the same way, and only recently have we started doing what we call genetic engineering on corn. So I want to talk about corn for a little while so you can kind of get a feel for how corn came to be what it is, and how we know that it is what it is. One of the things that I've been impressed by during the COVID crisis is people see scientists talking about things, and scientists say, this is what we know, but scientists don't ever do a very good job in many cases of saying, this is how we know it, and this is why we think that the thing that I'm telling you right now is the correct thing. Thing that came up a couple of days ago while I was on a bike ride is I overheard a conversation with two of the guys that I ride with. One of the things about bicycling is you're united by bicycling and by nothing else. There's a lawnmower outside my office. So you're united by bicycling and nothing else. And so you find yourself biking with people who oftentimes have very different backgrounds than you and very different political leanings than you. And so I heard these two guys, and one guy said to the other guy, did you see that story about the fact that COVID was designed in a Chinese lab and sent to America for whatever nefarious purposes? Well, this is an idea that's gone around the internet, and some scientists have actually studied the evolutionary history of coronaviruses, and the coronavirus that we're currently dealing with has all the hallmarks of a coronavirus that jumped from one animal out in nature to humans, because in many cases humans eat bushmeat. And so when you eat bushmeat, you're prone to get the diseases that are in those animals that you're eating. And there are characteristics in the genome of organisms that have been altered through genetic engineering that you can find as you look at the DNA sequence of things, or RNA sequences, depending on the type of virus that it is, that indicate that it's a natural virus rather than some virus that has been altered. And so scientists could explain this to people if they took the time to, but oftentimes you don't have the time to do this. And so one of the things I want to do in this class is to explain why we know the things that we do, so that when I say we know X, you can have some faith in the fact that we actually do know X. whatever X is. So we know that corn, modern corn, came from teosinte. And so I was looking over this in preparation for today. So one of the ways that we know that corn came from teosinte is we have archaeological evidence. If I was not a biologist, I would probably have been an archaeologist. And so do you guys know what a midden is? M-I-D-D-E-N? Middens are fascinating to me. A midden is an ancient trash heap. So wherever humans have been in their history, they create trash and they pile it up somewhere. And so if you go, for example, out to the Pacific Northwest, there are a whole series of fascinating papers that have been written about middens of Native American groups in the Pacific Northwest. And in those middens, you can see a long history of their food utilization. And so if you dig down in a midden, so a midden is just, they end up being mounds. And as you start digging down into the midden from the top, the most recent trash is at the top, and as you dig down through the midden, you are basically going back in time. And in the Pacific Northwest, there is a particular time period, and below that time period, the midden is full of salmon bones. And after that time period, in the upper portions of the midden, the degree to which salmon bones are found in the midden goes way, way, way down. And it turns out that this boundary line in these middens corresponds to the rise of salmon fisheries, commercial salmon fisheries in the Pacific Northwest. And when archaeologists started digging through these middens, it was very clear and obvious when you moved from pre-commercial fishing of salmon to post-commercial fishing of salmon. Well, in the same way, you can go to Mexico and Central America, and you can dig into middens that occur there. And what you find is that as you go back in time, corn got smaller and smaller and smaller, which means that over time, corn has been increasing in size, both in the length of the ear, the diameter of the ear, and the number of kernels on the ear, and also the relative size of the kernels on the ear. So the grains themselves have gotten bigger over time. The cob has gotten bigger, both in length and in diameter. And so this represents an evolutionary change over thousands of years, about 9,000 of years that corn has been under cultivation. This is evolution of the cob, of the ear of corn. This shows you a drawing of what teosinte looks like in terms of its growth form versus modern corn. And then this is the seed head, the ear of teosinte and the ear of modern corn. You can see that teosinte is a multi-branched structure. It has male flowers at the tip and female flowers along the branch, but it differs substantially from corn in that corn has a single main branch and usually one or two side branches as opposed to multiple side branches. And on each side branch, there's just a single ear of corn as opposed to multiple ears of corn on each branch of teosinte. When you look at the ear itself, you've already seen the picture of teosinte. You see that there's just a single row of kernels of grains, and those grains are relatively small. And maize has multiple rows of grain on a much larger ear of corn. And so these changes have been wrought through the process of artificial selection. Now, as it turns out, you can still breed this corn plant with this teosinte plant. So after all of this time of artificial selection, these two plants are still not reproductively isolated from one another. You can take modern corn and breed it with teosinte. And this is actually what Yang et al. Did in 2019. Now, one of the advantages of doing that is you can now create a hybrid, basically taking corn and teosinte, breeding them to one another, getting that hybrid, and then allowing that hybrid to breed with itself to cross-fertilizing among those hybrids. And from this, you can begin to dissect what the genetic components are that have resulted in the transition from teosinte to maize. And so the next slide basically starts to get into that. So these guys that published this paper in 2019, they looked at a bunch of different traits. Some of these traits are vegetative. Some of these traits have to do with responses to the environment, and some of these traits are reproductive. I want you to concentrate for now on the reproductive ones. So the reproductive ones are the ear length, the number of grains per row, the ear diameter, the grains per ear across the entire ear, the length of the plant from one node to another node, which is less important, but the total number of grains per plant and the total grain weight per plant and grain weight, the actual size of the corn kernels themselves. So these are the traits that they looked at. They all have their own abbreviations, but what you'll notice here is that all of these things from ear length to grain weight occur in this section of the chart down here. So when we think about how traits are determined, there are two forms of genetic variation that are of interest to us in this example here. One form of genetic variation is additive genetic variation, this thing that they show VA, which is the red bars. The other one is dominance variation, which is in the green bars. Now, I want to take a minute to explain what we mean by that. And I mentioned it earlier, but it probably slid past you, so I'm going to go back and reiterate it. These kinds of traits from Mendel's work, where you have a dominant allele and a recessive allele, and the dominant allele always gets expressed, and the recessive allele only gets expressed when it's by itself. Otherwise, it gets masked by the dominant allele. So that when you breed a tall plant to a short plant, all of the offspring end up being tall. And there's not an intermediate sized plant. You're either tall or you're short. There's no medium in here. So we don't get tall plant, intermediate plant, and short plant. We get 3 tall plants and we get 1 short plant. When you see the term dominance variance, or that VD, it's talking about this kind of gene action. These either on or not sorts of genes. It's either tall or it's short. It's either green seeds or yellow seeds. That's dominance variance. Additive variance is this situation here, a situation where you have traits that are determined by lots of genes, each gene contributing a little bit to the phenotype of the organism. We analyze these genes with quantitative genetic methods. We have continuous variation in the traits. We don't just have NBA basketball players and the rest of us who are really short. We have NBA basketball players who are, what's the tallest position on a basketball team? Any basketball players?
The center usually.
Oh, so we have centers, point guards. Who's shorter on a basketball team?
Your guards.
Yeah.
Okay, guards. So we have centers and guards, and then we have the rest of the population who is not in the NBA, and then we have shorter people. But there's this continuous variation. That's what we refer to when we talk about additive genetic variation. When we basically say, okay, well, I want to create a taller population of organisms, so I'm going to prevent short individuals from breeding, we increase the height of the parent population, which also increases the height of the offspring population. Those kinds of changes are gradual. It takes time for those to occur. But a lot of times, if we exert selection against a trait that is largely made up of dominance variation, those changes are relatively quick because we very quickly get rid of all of these recessive alleles, only the dominant alleles remain, and that's where we get the trait that is desired, in this case, a taller plant. Well, as it turns out, when we look at panel B down here, it's basically showing you of the amount of the total phenotypic variation that is due to genetics, how much of that is due to additive genetic variation versus dominance variation. And when we look at the reproductive characters in particular, we see that there are a couple of these, a small number of these, where a big chunk of the variation in the plant is due to this dominance kind of variation, this either or kind of variation. And those things have to do with the total grains per plant, the total grain weight per plant, grain weight, grains per ear, and ear length, and also ear diameter. So 1, 2, 3, 4, 5, 6. There are these 6 traits that they have an additive genetic component to them, but they also have a substantial dominance component to them. One of the important things about that is those traits then are easy for people to see. And when you choose a plant that has, you choose a plant to breed that has more grains per plant, chances are you're choosing a plant that is homozygous dominant for whatever that characteristic is. And as a result, evolution will occur relatively quickly. And so they discovered all of this by basically crossing teosinte with maize. And these traits were not particularly dominated by dominance variation in teosinte, but over evolutionary time, they have become more and more governed by these genes that are dominance related genes. And so not only have we changed the phenotype of corn over the time that corn has been in domestication, we've also changed kind of the proportion of the genetic variation for those traits that is dominance versus additive. And a lot of that has happened actually in the characteristics that have to do with the structure of the ear of the corn, which of course is the part of the corn that we consume. And so corn is a good example of a genetically modified organism that has undergone modification largely through selective breeding that has changed it from the structure of teosinte to the structure of modern corn. And it's happened through the manipulation of a relatively few number of genes that the people who were doing the selective breeding didn't even know existed. They were just trying to get more grain out of each plot of land that they planted. And as a result of that, they inadvertently selected for certain characteristics of the corn. Those certain characteristics made the corn more productive. And they happen to be genes that were selected that are these kind of genes of large effect rather than genes of small effect. Additive genetic variance is genes of small effect. Dominance variation is genes of large effect, is one way of thinking of those things. Questions about that? I just loaded a ton of fairly complicated genetics on you. Any questions? Anything that's unclear? All right, there's a lot of other stuff that's in that paper that we're not going to concern with because it doesn't really have as much to do with the relevance of what we're talking about today. So now when we come back to this meme, what are your opinions about this meme now?
Wouldn't it be that it's true?
It is true, isn't it? It's genetically modified. Woo! Are you concerned about that?
No, it's one of those buzzwords that's used nowadays.
It is. It's a meme that is trying to scare you by presenting, oh my gosh, what happened to that thing that corn came from? But really, we got to this particular form of a corn cob just through the same selective breeding that led to a pug coming from a wolf. Quite frankly, I'm way more disturbed about the pug because the pug has trouble breathing. My sister has a pug, and the pug just doesn't seem happy because he can't breathe. So I'm way more worried about the pug than I am about the corn. So that's what, when I talk about genetic modification, that's what I'm talking about. Selective breeding over long periods of time that lead to, in some cases, major phenotypic changes. That is different from genetic engineering. And so now we're going to spend quite a lot of time talking about genetic engineering. I'm going to begin by talking to you a little bit about the history of genetic engineering. How long have we been doing this? What kinds of traits have we manipulated? What proportion of our food supply is made up of genetically engineered crops, etc. Then we'll go into the specifics of, okay, this is what genetic engineering is in our food supply. What exactly is happening that creates a genetically engineered organism? And then that will hopefully equip you to deal with the things that are in the National Academy of Science report. Chapter 3 of the National Academy of Sciences report has a lot of the same information in it that I'm going to present to you today. I've just kind of boiled it down, and I'm going to try and do it in a way that is a little more relatable to non-scientists, although we'll see how that goes. So there are plenty of genetically engineered crops. We have alfalfa, canola, from which we get canola oil, corn, which we eat, and we also get corn oil from cotton, papaya. I was surprised when I learned that papaya is actually has actually been genetically engineered. Potatoes, soybeans, squash, sugar beets, and tomatoes. And tomatoes were actually the first organism that was genetically engineered. So we can think about it just in terms of what crops are genetically engineered, but we can also think about, well, what proportion of crops are genetically engineered versus non-genetically engineered. And this is from your textbook, if I recall correctly. And so whenever you see GM, GM means genetically engineered, non-GM means not genetically engineered. So this is another one of those cases where I would rather that books were a little more careful with their terminology, because non-GM versus GM, all of these non-GM crops have undergone some form of artificial selection, genetic modification, as opposed to genetic engineering. So when we look at two big crops, so in this class, we haven't done this a lot, but now we're going to start talking a lot about corn and soybeans, because corn and soybeans makes up a super huge chunk of what we eat. And so it's important that we know about corn and soybeans. So this shows you kind of all the hectares that are planted in soybeans, way more than half of them have been genetically engineered for at least one trait. And that's a fewer proportion of the total hectares that have been planted for corn. But when we look at the number of traits that have been engineered in corn versus soybeans, relatively few traits have been modified in soybeans, mainly herbicide resistance, and we'll talk about what that herbicide resistance is. But there have been many more different kinds of genes that have been altered in corn. So of this little tap of corn that has been modified, how much of that is due to herbicide resistance, insect resistance, or resistance to both? So there's been a wider variety of modifications to corn than soybeans. So, whoa, I just hit my mouse. So kind of a history. It goes back a long time. When were you guys born?
'99.
Oh, 99. Okay, so you've been eating genetically engineered crops your entire life. So in 1982, the first GM plant was produced, and this was a kind of a test of concepts idea, in that they put antibiotic resistance into tobacco. And in 1986, they put herbicide resistance into tobacco. And then in 1987, they took a thing called BT and put it into tobacco. So let me tell you what some of this means. I can now get rid of this genetics because we're done with that. So whenever you see the term BT, so BT corn or BT soybeans or things like this, BT stands for a bacterium. Oh man. I had it in my head what the name of it was, and then I lost it, and then I got it back. It's a Bacillus strain, thuringiensis. Whenever you write scientific names, you should either write them in italics or underline them if you're writing them by hand. So Bacillus thuringiensis is a species of soil inhabiting bacterium. And this soil inhabiting bacterium produces a toxin, and that toxin is toxic to insects. So what scientists did was they took the gene that is responsible for producing the toxin in the bacterium, they cut it out of the bacterium, and they inserted it into the genome of the tobacco plant. And as a result of that, now the tobacco plant produces its own kind of innate insecticide, essentially. And so the farmer doesn't have to spray pesticides on its crop because the tobacco itself is producing a form of pesticide. In a similar way, there's an herbicide-resistant gene that allows plants to withstand the application of herbicide. The most common of these herbicides that we have resistance to is called glyphosate. This is the scientific term for it, but you see this as the main ingredient in Roundup. So if you have ever used Roundup on dandelions in your yard or weeds, you know, coming up through the cracks in your driveway, it's Roundup uses glyphosate. And so one of the most common herbicide resistant traits is a resistance to this particular herbicide. The advantage of that for farmers is that you can spread herbicide on your field and it will kill all of the weeds, but it will leave your crop standing because your crop is resistant to the herbicide Roundup. And so one of the questions that you might have is, why were they doing this with tobacco? And the reason that they were doing this with tobacco is that the first mechanisms for moving genes around, what they used was a virus that already was known to infect tobacco. So they did this first using a thing called the tobacco mosaic virus. It's a virus that was already hijacking the genome of tobacco, and so that served as a good vector for doing this. And so that's kind of why the early history is so tied to tobacco. It isn't that tobacco farmers needed these things. It's just that when you're developing a new technology, you oftentimes go with the easiest thing to do first. By 1994, the Flavor Saver tomato had been developed. This was a tomato that had delayed ripening, so that you could grow a bunch of tomatoes on a farm, you could harvest those, and you could get them to market, and they didn't ripen so quickly so that they didn't ripen in transit and then get to the market as a mushed up ball of tomatoes as opposed to a bunch of nice healthy tomatoes. And so that was what the Flavor Saver tomato was about, first approved for sale in 1994. By 1995, we had potatoes that had Bacillus thuringiensis toxins put into them. That's the first insecticide producing crop to be approved, and it was quickly followed by canola, corn, and cotton later that same year. By 1997, we had herbicide-resistant wheat. Monsanto applied for a patent on this, and they were going to market this to Japan, but then Japan started worrying about genetically engineered crops, and so they basically do their best to not import any genetically engineered crops to Japan, and that caused problems with Monsanto's business plan to a certain degree. 2005, perennial genetically engineered crops approved in the U.S. One of the nice things about most of these crops is they're annuals. You plant them, they grow up, they produce fruit, and then they die. It's more problematic potentially if you're doing this with perennials because they persist on the landscape for longer periods of time, which means that there's more possibility for these genes that you've put into them to escape out of the crop that you've put them into and escape into natural crops. So that was an herbicide-resistant alfalfa strain. And then 2013, by 2013, 87% of soybeans that are produced are genetically engineered. And after complaining about other people's poor use of genetically modified versus genetically engineered, I look at my slides and I see that I'm using the GM label to refer to this. I'll go back and change that and update the slides. I'm talking about all of this being genetically engineered. So you can look at a graph, and this is from the Agricultural Survey, which is once again, it's a USDA product. One of the things, once again, that I like about government databases is that the data in government databases is put together by career scientists rather than political appointees, and the career scientists view their job as getting the best information to policymakers as they can. And as you can see, starting in 1996, the percentage of planted acres in all of these crops has just been steadily increasing. That increase was fastest with soybeans, but it's been similar trajectories for all of these other different crops. So to think that you're eating crops, that you're eating food that has not been genetically engineered, if you think that, you are living in a dream world, because a lot of our food is generated from corn and soybeans, and large proportions of corn and soybean has been genetically engineered in one way or another in the United States. These figures back here were globally, and so corn and a lot of other parts of the world are not genetically engineered. And when you get to the chapter in the National Academy of Science report, you're going to see why that's the case. These data here are for the US. And so US farmers, what basically happens is US farmers have more money, genetically engineered crops are more expensive, but they are willing to pay that premium because they produce higher yield. People in third world countries just simply can't afford those crops that are genetically engineered. So we'll pick up here on Friday. Questions? All right, get reading, and I'll see you on Friday.